Hi, this is the fourth lesson for Chapter 5, where we're going to be looking at some mechanisms where the cell or organism placed in a solution with a different tonicity than its body can use energy to adjust for those differences compared to what's inside of its body. So these are mechanisms of active transport. We've previously talked about passive transport that does not require any energy to be expended for that cell or organism to make adjustments, but here it would require energy. So we're going to look first at an example of an organism called paramecium. Paramecium is a freshwater protist. It's a single-celled eukaryote, so it does have nucleus. Actually, it has two nuclei, macro and micro, but in addition, it has an organelle that we learned about previously called the contractile vacuole. And this is a really interesting organelle that acts kind of like a pump, where when the paramecium is basically bathing in fresh water, which it always is, water through osmosis is always going to be rushing into the cell, into the paramecium, because the fresh water around the paramecium is hypotonic compared to out inside the paramecium's body, it would be hypertonic. You can sort of tell that because of how much stuff you see in the cell inside of paramecium. So that is why the water is going into the paramecium. Now you might recall from the previous lesson where we talked about mechanisms of passive transport for a cell, when an animal cell or a plant cell are placed in a hypotonic solution, the water is going to go into that cell, and ultimately the cell could take on so much water that it bursts. And paramecium is more like an animal cell in that it only has a plasma membrane surrounding its body. And so that plasma membrane could potentially rupture, causing all of the organelles inside of the paramecium to just spill out into the water, and it ultimately would die. But this doesn't happen because of the contractile vacuole. The contractile vacuole takes on that water and then pumps it out. But that does require that energy be spent by the paramecium. There are other single-celled eukaryotes that have this organelle, but this organelle is pretty unique in that it's not found in many other types of organisms. So there are other mechanisms that can be used by a cell to actively transport materials across its cell membrane. So now we're going to zoom into what might happen at the level of the cell membrane involving the use of energy, which in the form of ATP. This is the molecule of energy that a cell can use to perform work. So in general, we're going to look at the illustration to the right here as a generalization of what happens for a membrane level active transport process. So notice that there's a higher concentration of the solute inside the cell here at the bottom compared to outside the cell at the top. And so what's happening here is you see in this, instead of time, this is like a sodium potassium pump that actually can take on two different types of molecules. But if you just focus on the one that's appearing to go through now, the diamond shape, the orange diamonds, they're moving from low to high concentration. So the only way that that's possible is with the use of ATP through active transport, because as you might remember, diffusion with no energy uses or transports a molecule from an area of high to low. So this is low to high, that's the opposite. So we need energy to go against that concentration gradient. But something that is in common with passive transport at the membrane level is that these proteins that facilitate the transport can only bind to specific molecules. Sometimes it's just one type of molecule. Sometimes they have receiving sites for two different kinds, like shown here. So let's take a closer look at this particular example, which is like a sodium potassium pump. which is responsible for how nervous impulses are transmitted throughout our body. We need the sodium-potassium pump to receive input, sensory input, and then have that information transmitted from our nervous system to various parts of the body. We're not going to get into the nitty-gritty of the sodium-potassium pump. Like, you don't have to know the steps here, but just recognize why this is an example of active transport. So the key thing to focus on here is where are the initial concentration gradients for the sodium and the potassium, and how are those molecules moving across the membrane, and is energy required or not? So first, let's notice that the sodium ions enter. They're moving from the cytoplasm, where there's a low concentration, and they're going, once all three molecules bind, they're being moved across the membrane into the extracellular fluid, where, although it's not indicated here on the third step, in the beginning it told us that it's high. So it's going from where you had initially low concentration sodium to high. So that is against the concentration gradient. Come on, text box. There we go. All right, so in order for that to occur, we needed some energy to go against the gradient, so that's why you saw in step 2 ATP. All right, then after sodium is leaving the cell, the potassium ions enter. As also shown in the initial state here, there was a low concentration of potassium to start with outside the cell, and a high concentration inside the cell. So if, again, those ions of potassium are moving from low to high, there needed to be a source of energy. So that's where the release of the phosphate group comes in. It was added on there when the ATP was used in step 2, so it's still related to the addition of the ATP, the energy molecule, but specifically it's the phosphate group that provides the source of energy through the breaking of the bond to open up that protein transporter and release the potassium inside the cell. And then the cycle continues. Pretty fascinating. We have another example of active transport that also can be found in the human body. This is the sodium glucose cotransporter. So like the sodium potassium pump, it does have binding sites for two different molecules, sodium and glucose. Now, however, differently from the sodium potassium pump, only one of these is being transported against the concentration gradient, and that is the glucose. We always need glucose. So even if it's still already high where it's going, your sodium glucose transporter will continue to pump the glucose across the membranes, building up even a higher gradient, higher concentration. So the energy that fuels this pump comes from the sodium, which is actually going down its gradient from high to low, like diffusion, but that energy from that is what's fueling the cotransporter. So the glucose cannot be transported unless there's a gradient of sodium to fuel that pump. Instead of ATP, this is how it's operating. However, there's still energy that is expended by the cell, and that's the big point of this part of the lesson. Now, these examples we just looked at we're zooming into the membrane level. We're seeing what's happening in that phospholipid bilayer. But sometimes molecules need to be transported across the membrane in bulk amounts, not just one molecule at a time like shown here, but in larger quantities. So the mechanisms of bulk transport of materials both in and out of the cell are called endocytosis and exocytosis, or elimination. And this representation of a cell, an animal cell, shows the whole kind of cycle of endocytosis and exocytosis, where a molecule of food is being engulfed, which ultimately becomes a food vacuole, surrounded by its own plasma membrane, which is now the membrane around the food vacuole, which then fuses with lysosomes containing digestive enzymes to break down that food. Some of that energy will be absorbed by the cell as the food, and then the waste that remains can be expelled or eliminated from the cell when that digestive vacuole fuses back with the plasma membrane. So now the lipid bilayer's recycling. Wherever it was like used to make a vacuole, it's going to end up going back to the outer plasma membrane, and then the wastes are released. Like the active transport processes that we saw at the membrane level, this also requires energy. This is also active transport. It's just when you have bulk amounts of materials being transported in and out of the cell. Now, just like us, the cell, certain types of cells that undergo these processes can take in large quantities like food, eating. So phagocytosis is a type of endocytosis known as cellular eating. So like shown here, the cell is engulfing a large molecule of food, but there's other smaller materials in the solution around the cell that could also be utilized as a source of nutrition for the cell. So that would be pinocytosis, or cellular drinking, taking in some of the solutes that are in the fluid and forming similarly a membrane-bound organelle, but they're usually smaller. And so the smaller membrane-bound organelles that form by the cell's own plasma membrane are called vesicles. So a vacuole will be bigger, vesicle will be smaller. We're not going to be too concerned about the coatings on the vesicles. That's involved in the cell-to-cell communication, a topic that we don't really dive into in this class. So for now, just know the difference between phagocytosis, pinocytosis. And either one ultimately will result in exocytosis, where that vacuole or vesicle will fuse back with the plasma membrane of the cell. And that's an important thing, because if it didn't, the cell would ultimately get smaller and smaller and smaller as it keeps forming vacuoles and vesicles that become part of its interior organelle system. That would ultimately cause the cell to keep shrinking, 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 shrinking. So it's important that those vesicles and vacuoles do fuse back to the outer plasma membrane so that the cell can maintain its size and continue to exist. That concludes our lesson on Chapter 5, and actually all of the lessons on Chapter 5 are now completed.